In a previous video, I started to discuss the connections between the level of body fat and health. As you recall, an healthy population should have an amount of body fat in a normal range, as shown on this graph, with few individuals having too little or too much body fat. We can learn a lot about the regulation of fat storage by studying diseases associated with a level of body fat outside of the normal range. As described in a previous video, diseases such as lipodystrophy and cachexia are associated with an abnormally low level of body fat. In this video, we'll explore what happens at the other extreme, when there are too many lipid droplets, and I'll focus on two types of diseases: neutral lipid storage disease and metabolic syndrome. This image shows the distribution of fat in an healthy individual. Fat is distributed in the subcutaneous tissue with quantitative differences that are influenced by age and sex. Adipose tissue is also found around internal organs. As long as the fat storage stays in the adipose tissue and is not excessive, your body functions healthily. Health problems arise when caloric intake is not balanced by an appropriate energy expenditure. This unbalance leads to obesity and correlates with fat storage overflowing into tissues and becoming toxic because these tissues are not equipped to handle an excess of unprocessed fat. This process is called ectopic fat deposition. Let's describe different diseases associated with ectopic fat deposition. Neutral lipid storage disease is a rare genetic disease resulting in the body's inability to correctly store lipids. To understand this disease, we first need to look at adipocytes, the cells that make up adipose tissues. Mature adipocytes are cells that each contain a single, very large lipid droplet. The size of the droplet is so large that the cytoplasm is reduced to a thin layer at the periphery of the cell. The cell nucleus is pushed against the cell membrane. The lipid droplets in adipocytes are filled with triacylglycerol, the major form of energy storage and insulation. Neutral lipid storage disease is considered as a monogenic disease because it is associated with mutations that affect a single gene. There are in fact two forms of this disease. The first one is characterized by a type of heart disorder called cardiomyopathy. This subgroup is called NLSDM. The second type of neutral lipid storage disease is characterized by ichthyosis, where the skin is dry and rough like fish scales. This subgroup of neutral lipid storage disease is called NLSDI. So let's take a look at NLSDM first. This subgroup of neutral lipid storage disease is associated with mutations in the gene coding for the lipase ATGL. As you recall from a previous video, ATGL is an enzyme required for lipolysis and is active when recruited at the surface of the lipid droplet. ATGL is one of three lipases that convert triacylglycerol into free fatty acids and glycerol molecules. The other two lipases are called HSL and MGL. ATGL catalyzes the first step of lipolysis, that is the conversion of triacylglycerol into deacylglycerol and a molecule of free fatty acid. Mutations in ATGL prevent its recruitment to the surface of the lipid droplets. So lipolysis is impaired. The patient cannot use its fat storage in times of need. And if caloric intake continues to increase, the adipocyte exceeds its triacylglycerol storage capacity, leading to ectopic deposition of fat and lipotoxicity. How exactly does disrupting the function of ATGL lead to cardiomyopathy? Fatty acids released by lipolysis are needed for proper cell signaling and gene expression in mitochondria. ATGL defects result in oxidative phosphorylation and energy production impairments that affect tissues such as the cardiac muscle that are highly dependent on normal mitochondrial functions. In mice, studies have shown that agonists that restore the defective signaling pathway can reverse ATGL-dependent mitochondrial defects. These agonists serve as a starting point for the development of drug treatments in patients suffering from NLSDM. Now, let's look at the other form of neutral lipid storage disease, NLSDI. In the 1970s, two physicians, Charanin and Dorfman, did a detailed study of NLSDI. That is why this disease is also called the Charanin and Dorfman syndrome, or CDS. This form of NLSD is associated with mutations in the gene coding for the alpha-beta hydrolase domain-containing protein, or ABHD5. So what's the function of ABHD5? As you recall, this protein is known to activate ATGL. Therefore, you would expect that NDLS-M and NDLS-I patients would share common symptoms. However, it's not the case. NDLS-M patients have cardiomyopathy, but do not display ichthyosis, while it is the opposite for NDLS-I patients. So these observations suggest that ABHD5 has a role in triacylglycerol regulation independent of ATGL. This role has not been yet characterized. So what is ichthyosis? This word comes from two Greek roots, ichthyo meaning having to do with fish, and osis that means pathology or abnormal condition. The name reflects the aspect of the patient's skin, dry and rough like fish scales. Upon closer inspection of the patient's skin, scientists found an abundance of lipid deposits. Patients with ichthyosis lack proper skin permeability. Your skin has what is called a permeability barrier. Let's think about it with a brick and mortar analogy. The bricks in the skin's permeability barrier are made of specific structural proteins. The mortar holding the structural proteins is made of a lipid-based extracellular matrix. The matrix is made from specially processed fatty acids that come from the lipolysis of triacylglycerol. The combination of structural protein and the lipid-based matrix makes a strong wall protecting us from the outside environment. If ABHD5 is normal, the permeability barrier remains intact and functions normally. In NLSDI patients, the ABHD5 mutation blocks the proper lipolysis, leading to an abnormal triacylglycerol deposition in the skin, to a lack of processed fatty acids, and ultimately to a disruption of the lipid-based matrix. The consequence is a breakdown of the mortar holding the bricks. The result is a skin taking on a dry, rough, and scale-like appearance. There is still much to learn about the ATGL-independent role of ABHD5 and about the apparent skin-specific effect of ABHD5 deficiency in NLSDI patients. In the last part of this video, I'll focus on metabolic syndrome. Metabolic syndrome is characterized by a cluster of pathologies, including high blood level of triacylglycerol, cholesterol, and LDL, hyperglycemia, and insulin resistance. It is considered as a polygenic disease. What does it mean? In contrast to neutral lipid storage disease, where a single gene is mutated, metabolic syndrome is associated with mutations affecting a group of non-allelic genes. The pathologies associated with metabolic syndrome are correlated with obesity and high calorie intake. Even if a patient is not consuming fat, excess of carbohydrates and amino acids eventually are converted into fat. The body tries to answer to this excess fat storage in two ways: by increasing the size of existing adipocytes and by increasing the total number of adipocytes. Conflicting evidence has led to a debate of the role of this excess of adipose tissue. Some argue that the increase in adipose tissue has a protective role. The body tries to protect itself against the negative effects of metabolic syndrome, including the toxic effect of ectopic fat deposition. Others argue the excess adipose tissue plays a harmful role and is one of the factors responsible for metabolic syndrome. It boils down to whether obesity is the cause or the effect of the onset of metabolic syndrome. Regardless of the debate, let's focus on the consequences of ectopic fat deposition. This ectopic fat deposition is strongly correlated with lipid toxicity and increased inflammation, and could trigger insulin resistance and finally the onset of type 2 diabetes. So how does this fat deposition occur? Studies suggest that there is a genetic component. As you recall from a previous video, perilipin is a protein attached at the surface of lipid droplets. Its phosphorylation results in a change of conformation, allowing access to stored triacylglycerol by lipases. In other words, perilipin controls the size of lipid droplets. Studies have shown that levels of perilipin strongly correlate with insulin resistance. This suggests that perilipin might aid in preventing lipotoxicity. Excess of triacylglycerol and larger adipocytes can also lead to chronic inflammation. This part of the figure represents normal conditions. The adipocytes are small, and triacylglycerol intake is compensated by an equal degradation of stored triacylglycerol. The adipocytes have not reached their storage capacity. A normal amount of fatty acid produced by lipolysis is exported in tissues such as the muscle. The uptake of glucose by muscle is normal, and mitochondria produce an expected amount of ATP. When an individual becomes obese, the fat intake surpasses the amount of degraded triacylglycerol, and adipocytes become larger. The enlarged adipocytes secrete a chemokine called MCP1. This protein recruits cells from the immune system that are called macrophages. Macrophages infiltrated into the adipose tissue in turn secrete a protein called TNF-alpha. This factor increases fatty acid export to muscles. Excess of fatty acids in the muscle lead to an ectopic deposition of these fatty acids that interfere with the targeting of glucose transporters at the surface of muscle cells. The result is a decrease in glucose intake. So even when insulin is secreted by the pancreas in response to a high blood glucose, the reduction of glucose intake by muscle cells leads to insulin resistance. So insulin resistance is defined as the inability of insulin to stimulate the transport of glucose from blood to tissues. While the pancreatic beta cells are fully functional and secrete insulin, your body's cells have lost their ability to respond to insulin. Insulin resistance can lead to type 2 diabetes, the topic of another video, and to cardiovascular diseases. While the mechanisms leading to insulin resistance are not fully understood, scientists believe that ectopic fat depositions have something to do with it. It is important to note that certain lifestyle choices, such as overeating and lack of exercise, are risk factors that exacerbate the metabolic syndrome. and contributes to obesity, insulin resistance, and eventually to type 2 diabetes. So in this video, we have looked at how genetic disorders are correlated with excess body fat and too many lipid droplets. We saw how mutations in ATGL and ABHD5 could lead to neutral lipid storage disease, and how metabolic syndrome can have both external and genetic components. We hope you will keep these things in mind before biting in your next hamburger or drinking a very sugary drink, whether it's a soda or an apparently healthy glass of orange juice.